helping to extract complex processes governing chemical polymorphism in biosystems. Biosystems are characterized by high complexity, which need to be analyzed according to different aspects, including regulation levels, variation ways, processes scales, and dynamics. Regulation levels in biosystems are governed by binary or dichotomous mechanisms, making a whole input resources to be competitively distributed into different compartments, leading to different proportions of different regulation profiles. The set of all the regulation profiles in a population is skewed from different expression or different resource distribution ways, generate several phenotypes or several chemical or several polymorphic trends, leading or attributing to biosystems a multidirectional aspect. Although the phenotypes represent final development states in populations, they initially result from locally interactive processes occurring in complex biological networks. These enclosed processes going from local to global scales attribute to the biosystems multi-scale aspects. Finally, regulation processes and resulting phenotypes show different regulation ways in time, or regulation laws in time. By this way, the dynamics and kinetics constitute a fundamental dimension for integrative analysis of biosystems. During my conference, I will present to you a new computational approach taking into account these four integrative aspects. The new computational approach starts from highly noisy data set, which is initially classified into different phenotypes. Then, variability between and between phenotypes are statistically combined to converge to a deep population backbone, from which polymorphism regulating mechanisms can be graphically highlighted. The simplistic illustrative case will consist of a chromatographic and pharmacokinetics dataset, including 4 metabolites, which were analyzed at different times in several patients in the blood of 34 patients suffering from Parkinson's disease. Parkinson's disease is not the subject of this conference, but it is just an illustrative case. The 4 metabolites concerned the level of dopamine or L-DOPA, which is the administrative drug, and 3 derivatives, including DOPA, HPA, and 3-ONE. For each patient, 7 chromatographic profiles were obtained for 7 hours following the L-DOPA administration. In all, the total number of chromatographic profiles was equal to 248 by considering all the sampling times and in the 34 patients. In the first competition step, this data set was subjected to a correspondence analysis, which highlighted different metabolic trends, which were characterized by high relative levels of some metabolites. Then, the 284 metabolic profiles were classified into appropriate metabolic trends by using or by applying the cluster analysis based on distance calculations between individuals. To start, application of correspondence analysis highlighted 4 metabolic trends which were organized along a gradient, opposing high regulation levels of 3-OMD to a high regulation of L-DOPA. Metabolite 1 was dominated by 3-OMD. Metabolite 2 was showed intermediate state with a lower regulation of 3-OMD in presence of L-DOPA. Metabolite 3 was characterized by high regulation of HBA, and metabolite 4 was dominated by L-DOPA. By referring to the metabolic chart of L-DOPA and its derivatives, metabolite 1 seemed to be favored by a short regulation pathway specific to 3-OMD. Moreover, this metabolite has a long half-life of 17 hours, favoring its accumulation. Opposite metabolic trend, represented by metabolite 4, was directly favored by L-DOPA administration. It showed the highest metabolic regulation, or it showed the highest metabolic ratio between L-DOPA and 3-OMD. Next metabolic transformation of L-DOPA will lead to an intermediate state represented by metabolite 2. Further and later transformation of L-DOPA will favor the development of HBA, the final product, which leading to the development of metabolite 3. The next computational step will consist in analyzing how these 4 metabolites quantitatively varied the most relatively to the others to favor multiple development of polymorphic background from a unified population metabolic background. Population metabolic background can be conceptually defined as the gravity center of all the possibly expressed metabolic profiles. Statistically, this can be carried out by barycentric calculations between all the metabolites by gradual variable weights. These combinations were applied by applying the shift mixture design, which provide a complete set of mixtures containing each one n individuals issued from Q groups. In our case, Q was equal to 4 metabolic groups or 4 metabolites. The number n represents the total and constant weight of each mixture to which the different groups contribute through different proportions given by the Shakespeare matrix. By fixing n to 10 individuals per mixture of 4 groups, combinatorial calculations depending on n and q give a total of 286 mixtures necessary to be carried out to cover the whole mixture design. These 286 mixtures, combining 4 weighted groups, can be geometrically represented into a tetrahedral simplex space containing 286 points with 4 coordinates. The corner points represent the pure phenotypes, and the edge, height, and inside points represent hybrid situations combining 2, 3, or 4 phenotypes respectively. This organization provides a multi-layer and multi-directional way favoring the development of chemical polymorphism by gradual weighting or by linear combinations between phenotypes. Application of the mixture design was carried out by randomly sampling 10 individuals for each mixture, issued from the 4 groups by considering their weights given by the Shakespeare matrix. in the output for each mixture, the 10 individuals were used to calculate an average profiles, memorizing the weighting effects of the 4 metabotypes on the chemical polymorphism of population. With 286 mixtures, we obtained a response matrix containing 286 average metabolic profiles. At this step, it is important to underline that each average metabolic profile was calculated from only 10 randomly sampled individuals, making the within group variability to be poorly explored. To overcome this variability deficit, the mixture design and its elementary response matrix need to be iterated several times. For that, bootstrap technique, which is a resampling technique, was applied, consisting of several samplings of 10 individuals for each mixture. At the end, the mixture design and its elementary response matrix were iterated 50 times. The number 50 was tested to be sufficient for results convergence. Finally, the 50 elementary response matrices, containing each one 286 average metabolic profiles, were averaged to obtain a unique response matrix containing 286 smoothed metabolic profiles. This smoothed matrix was used for graphical visualization of the relationships between metabolites in order to identify regulatory processes governing the chemical polymorphism in the student population. Scatter plots visualized from the smoothed matrix highlighted a highly organized relationship between the 4 metabolites. This indicated that the computational approach converged to a deep population metabolic backbone. Moreover, spiral correlations calculated on the smoothed data on the hand and the experimental data on the other hand showed sign conservation for all the correlations concerning all the pairs of metabolites. This indicated that the computational approach was efficient to extract deep signals from initially highly noisy experimental data. Among the 4 metabolites, P, O, and D showed strong negative correlations with all the metabolites. These negative correlations were compatible with the competitive metabolic pathway of 3-OMD and its cumulative concentrations by opposition to the rapid elimination of the other metabolites belonging to another allosynthetic pathway. HBE, however, showed lower compressed relationships with multidirectional aspect. This was compatible with the distant and closer location of HBA in the metabolic network. Interestingly, these scatter plots with the lower linear trend and multidirectional aspect were very rich in information because they included several superimposed relationships depending on phenotype. To highlight this phenotype-depending relationship within the same scatter plot, the different weights of each metabotype were projected on corresponding points of the simplex space. Then, equal weight values were statistically grouped by a confidence ellipse. For each metabotype, we obtained 11 confidence ellipses, going from 0 indicating absence to 10 indicating the full development of phenotype. Finally, the succession of the 11 weight ellipses will define a trajectory showing how the 2 metabolites, in this case HBE and LOPA, varied to favor the development of considered metabotype. By this way, metabotype 1 revealed to be developed through a strong decrease of LOPA and HBE regulations. Also, metabotype 2 was developed through a decrease of both LOPA and HBE, but LOPA was not reduced to 0 by opposition to HBE. In the same scatter plot, metabotype 3, however, developed through a strong increase of HBE regulations, while LOPA was stabilized at intermediate opposition to the three previous ones, show the negative relationship between HDA and L-DOPA, for which L-DOPA reaches its maximal levels in presence of unfavorable variations for HDA. Besides this negative global trend between HDA and L-DOPA, positive inclinations of weight ellipses highlighted a positive local relationship between these two metabolites. In other words, we have a global negative trend, including a positive local relationship. This positive local relationship indicated that L-DOPA was systematically prepared or ready to favor the increase of HDA in next step. This could be implicitly indicated by transition between metabolite 4 reached in L-DOPA at the right and metabolite 3 reached in HDA at left. After highlighting metabolic flexibility and the static aspect, further analysis of metabolic regressions can be carried out by taking into account the time. For that, the 248 chromatographic profiles previously classified into 4 metabolic trends were further separated into 7 time ranges, going from 0, 1 to 6, 7 hours respectively. Then each data subset, corresponding to a time range, was subjected to an iterative experimental design, leading to a final smoothed matrix. From the 7 smoothed matrices, 7 scatter plots were visualized for each pair of metabolites. Finally, dynamical aspect was considered by following the 7 scatter plots along the discrete time axis, T1 to T7. By this way, HDA and its precursor DOPA revealed to be concerned with an anti-clockwise hysteresis indicating lagged progression of HDA by comparison to DOPA. This lag time for HDA could result from the presence of two competitive precursors giving HDA, 3MT and DOPA. These two precursors compete for the same enzymes for their biosynthesis from dopamine and their transformations into HDA. In conclusion, the simplex approach revealed to be a strong tool for statistical treatment of highly noised metabolic dataset. It showed high interest for nonlinear and dynamical analysis from its ability to extract multidirectional, multi-state, and time-dependent relationships, helping to highlight complex processes governing the polymorphism in biosystems. Beyond these metabolic data, the approach can be applied to other types of data, including NMR, genetics, or ecological data, to highlight the governing polymorphic systems. For more information on computational metabolomics and simplex approach, I invite you to consult some of my publications and my books. Thank you for your attention.
Questions for Nabil? Well, maybe I was wondering, did you try any other strategies for the mixing beyond the Shifa's approach? Did you try any other strategies? Yeah, for the mixing beyond the Shifa's approach?
I published this approach in ecology by mixing the stomacal content of a predator in terms of several phase. And I showed by this approach multiple foraging behavior in predator toward different phase. The approach revealed that the predator behaved by memorizing last phase to lead to capture next phase. In ecology. But I did not apply it in genetics because I haven't done.
Okay, so there are no more questions for Nabil, and maybe we'll thank him one more time and all the speakers for today's session. Just to remind everybody that there are some refreshments available, I think, outside now, and post the session.
But I repeat that the point was
fix it.
That's